Okay, cytoplasm. We're going to go over the organelles. The cytoplasm is all the material inside the cell except for the nucleus, so not including the nucleus. And it's made up of cytosol, which is the fluid portion of the cytoplasm. There's the cytoskeleton, cytoplasmic inclusions, and organelles. Right, because did we take notes over the structure of the cell yet? No. Yeah. We didn't get through them all. Same thing. The cytoplasm. There's cellular material outside the nucleus. Oh, wait, we just talked about cytoskeleton. This supports the cell but allows for movements like change in cell shape and movements of cilia. There's 3 proteins that make up the cytoskeleton. There's microtubules, so just like the name says, micro means small, and tubules would be a tube, so they're hollow. And they're made up of a protein called tubulin. And their function is for internal scaffolding, transport, and cell division. Then microfilaments are made up of actin, and the microfilaments, so here's the microtubule right here. And then the microfilaments are right here. And they're a lot smaller. It looks kind of like, and we'll look at actin when we get to muscles, but it's kind of like a pearl necklace that's twisted upon itself. Its function is for structure, support of microvilli, contractility, and movement. And then the intermediate filaments look kind of like cables. They're long strands twisted, and they're for mechanical strength. Then the cytoplasmic inclusions are aggregates of chemicals such as lipid droplets and melanin. So basically little suspended chemicals like lipids inside. Organelles are small specialized structures with particular functions, and most of them have their own membrane that would separate the interior of the organelle from the cytoplasm. And they have specific functions. So first is nucleus. Nucleus does have its own membrane. That membrane is called the nuclear envelope, and there's an inner and outer membrane with pores that allows mRNA to get out. So do you guys remember what RNA does? What? Right. It's the DNA. DNA gets transcribed into RNA, and then the RNA is what the ribosomes attach to make proteins. The nucleoplasm is the substance inside the nucleus, and the nucleolus is that dense core. Where much of the DNA is located here. There's a little bit in the mitochondria, and the rest of it's located in the nucleus. The chromosomes are where the DNA, that's actually the DNA that's condensed. So there's a couple different ways. Most of the cell's life, it's as a chromatin, which is this long strand, and there's some proteins that it wraps around, but it's unwound in this DNA, the chromatin. But during cell division, that's where they condense, and you get a pair of chromatids. So this would be a chromatid, this would be a chromatid. Together, it's called a chromosome, and you're probably more familiar with seeing DNA look like a chromosome. And then each pair of chromatid is joined here at the centromere. So the chromatin condenses into a chromatid. Those sound really familiar, really similar. Chromatin condenses to chromatids. The pair of chromatids join at the centromere to form the chromosome. Questions on the DNA, I guess, before I go on? Ribosomes are sites for protein synthesis. There's a large and small subunit. So in this picture, you can see the small subunit. Whoops. Small subunit here, and then there's a large subunit, and they come together and surround the mRNA, and that's where the protein is going to be produced. And there are free ribosomes and attached ribosomes. Free ribosomes are just floating around in the cytoplasm, but attached are attached to the endoplasmic reticulum. And when there are attached ribosomes to the endoplasmic reticulum, those are called rough endoplasmic reticulum or rough ER. So proteins are produced there because it has the ribosomes. Smooth ER has no ribosomes and it manufactures lipids. And then there's the cisternae, which are the inner space isolated from the rest of the cytoplasm. So this blue portion right here would be the cisternae. It's basically hollow. Well, it's filled with some fluid, but it's inside and separate from the cytoplasm. And the proteins are produced by these, can't really see them very well, but there's little blue dots. Those would be the ribosomes. And the proteins would be produced inside the rough ER then and travel through, and then it would create a vesicle to go to the Golgi apparatus. And the Golgi apparatus, in biology, I think you learn it as like the UPS store, right, where it modifies, packages, and distributes the proteins and lipids. And they look like flattened membrane stacks stacked on each other. So it has its own membrane also. So in this picture, we have rough ER. Here's the ribosome. It's producing a protein. The protein gets produced, and it would create a vesicle. So it has its own membrane around this protein. So here's the ER in a cell. That vesicle is going to come and travel up to the Golgi apparatus, and it would bind to the Golgi apparatus, open up and empty the protein or lipid into the Golgi apparatus, where it's going to modify, if anything, and it continues to travel through the Golgi apparatus until it exits the Golgi apparatus as a secretory vesicle, and it's going to be secreted on the outside of the cell through exocytosis. Now sometimes the proteins are incorporated into the membrane, and they would be new membrane proteins, the channel proteins and stuff. Does that process make sense? Any questions on that process? Just goes through. So really, if you look at all the membranes, they're going from the endoplasmic reticulum to the Golgi apparatus to the cell membrane. All those membranes are the same. They're still a phospholipid bilayer because they're able to be interchanged and mixed with each other. Okay, a lysosome is produced, and it has digestive enzymes inside of it that would break down any of the molecules that it's brought into the cell. So if it's brought into the cell, it's called a vesicle, and then the secretory vesicles are secreted outside of the cell. And then the lysosome, and really, you can't tell the difference between a vesicle, a lysosome, and then a peroxisome. When you look at a picture, they look the same. It's just what the contents of that vesicle is what makes them different. So tomorrow when we start the lab, we'll be looking at the model of a cell, and you really won't be able to tell the difference between them by just looking. So as long as you know that it's one of the three, that's fine. But a peroxisome is smaller than a lysosome, and it has enzymes also to break down specifically fatty acids and amino acids, and hydrogen peroxide is a byproduct of that breakdown. And then mitochondria is a major site of ATP synthesis. What's ATP? Adenine triphosphate, and what does it do? Energy. Stores energy in the chemical bond, so when the bond breaks, it releases the energy to, we looked at yesterday to change the shape of the protein for that sodium-potassium pump. There's two membranes, and the cristae are the infoldings of the inner membrane, and then there's a matrix located in the space formed by the membrane. Mitochondria numbers will increase when cell energy requirements increase. So where do you think what kind of cells, what tissue would have a lot of mitochondria? Muscles. Your muscles require a lot of energy, so there's a lot of mitochondria in your muscle cells. Mitochondria is where there is some DNA located, and that codes for some of the proteins needed for mitochondrial production. So when cell division occurs, the mitochondria can produce its own will reproduce itself. Centrioles and spindle fibers, these are very important in cell division, which we'll talk about, well, after we do this next lab. And they're located in the centrosome, and it's just an area. There's no membrane around it. It just has, it's just the area where the centrioles are located. They would form microtubules, and during cell division, the centrioles divide and they move to the polar ends. Each end of the cell will have its own centrioles, and they will produce the spindle fibers that come over and attach to this chromosome, and it would pull the two chromatids apart during cell division. It looks like licorice. Do you guys remember? Now we'll go through this. Do you know which phase would be when they pull the centromere, this chromatin apart? Anaphase. Anaphase. PMAT. What's the metaphase? What's special about metaphase? How you can tell if a cell is in metaphase? The centriole, the chromosomes are lined up on the pole, on the equator of the cell. So when you see the chromosomes lined up in the middle, that's metaphase. And we will talk about the next time we have notes, we'll talk about mitosis. We don't do meiosis. It's meiosis. It's basically the same thing as mitosis. It just, yeah. Cilia, appendages projecting from cell surfaces. There's not really any cytoplasm in there. It's not. We'll see the difference between cilia and microvilli, but they're just appendages, and they're capable of moving. And it moves materials over the cell surface, and they're very common in the respiratory tract. So all this mucus that you produce in the respiratory tract, the cilia moves it up your respiratory tract until it reaches a point where you have a reflex to cough and cough it up. So you have the cilia is what's moving that mucus up the lining of your respiratory tract. What do you mean? What?
I've been thinking about that all period. She made a comment that really
Okay, flagella are very similar to cilia, but they're longer, and usually there's only one per cell, and it would actually move the cell. And it moves the cell in a wave-like fashion. So the cilia move substances across the cell, and the flagella would actually move the cell. Yeah. Microvilli are extensions of the plasma membrane, and its function is to increase the cell surface area, so there's more area for absorption to occur. Normally there's many on each cell, and they're 1/10 to 1/20 the size of the cilia, so they're very small and they don't move. So they don't move substances across, but it just increases surface area. This is what they would actually look like. Here, microvilli comes up and down on the surface of a cell. Where do you think this would be located? There's a lot of microvilli in a certain area in your body. No. Okay, you increase the surface area to increase absorption. Small intestine. Here. Yes, because you're absorbing a lot of nutrients and stuff. So there's lots of microvilli in your small intestine. Hey, grab your clicker. See how much you remember.
Okay, there's not enough people in here to really that representative. We're missing like 4 people. Now we're just like 6.
They're so tired.
You're never gonna see this.
Why is it after lunch we always get so tired of
No. No, you hook it up and it produces a stream of electrons, I think. It's supposed to. I've never seen one done in real life. The glass can start to produce X-rays,
Can we see it?
so we don't usually
Wait,
do
X-rays
it.
as in you stick your hand like on one side of it and you can see.
Oh, Mr. Stromer had it. I found it.
You got like a lead master.
Wait. So you're saying if it goes and you have your hand on the other side of it, you can see that?
If you had the film, I don't know, it's probably not enough.
Bounce stuff.
Yeah. You have to have the film to make it.
But we can still look at it.
So in other words,
Okay, which of the following cell organelles is not correctly matched?
Stop it.
Who's number 3? Because you haven't answered yet. This is the first time we've done this? Okay, the answer is, see what you guys said. C. Yes. Mitochondria. What do mitochondria produce? Energy, ATP, not proteins. What does produce proteins? Ribosomes. Yep.
You want to ask me why, but I thought this thing contains digestive system.
No.
Why?
Okay. Secretory vesicles are produced by secretes. What?
The secretory part.
This is Aubrey came to the drive-thru once last time. It was real weird.
Okay, what did you guys answer? It is A. Yes, the Golgi apparatus. So D. Okay, smooth endoplasmic reticulum produces vesicles, but those would go move to the Golgi apparatus, and then the Golgi apparatus is what produces the secretory vesicles. It's part of the chain. Which of these cells would most likely have the largest number of mitochondria? And, oh, I forgot to change the right answer. It is D, yes, or B, B. We talked about that one. Vesicles are formed by folding of the what? So different than secretory vesicles. Vesicles are bringing in substances.
Yeah, why? You were just like, I was like, are you gonna answer? Oh yeah, I'm gonna answer. No, but I know why mine's gray.
Answer is C. Yes. So you're bringing substances in through the cell membrane. Movable projections from the surface of cells found in abundance in the respiratory tract. Yeah. Oh, I did put on Edmodo and on Angel the copy of just the questions, since those weren't in the original notes that were up. Does everyone have it?
Yeah, me easily.
Answer is B, cilia.
I can never do it quick enough.
And that's it. So for the rest of the time, you need to work on your packet. So get your packet out.